Microbiology, as well as Dr. Bajrang Kumbar, postdoctoral fellow, IIT Mumbai. So it's now a great pleasure for us to welcome Dr. Rebecca Thombre, ma'am, who has chaired today's session. So we do welcome ma'am on the behalf of all our students and faculty as well as management. Thank you, Devi.
Thank you, ma'am. Yes, it's indeed a pleasure to welcome Dr. Rebecca Thombre, today's guest speaker. As it is said by Hillary Clinton, women are the largest untapped reservoir of talent in the world. So I think, forbidding the statement, Dr. Rebecca Thombre had proved to the world that it's not so. Yes, going ahead with the introduction of today's chief speaker, I request Dr. Tushar Borse, the coordinator of the Innovation Institutional Innovation Council, which is under the Ministry of Education, Government of India, to kindly introduce today's speaker, Dr. Tushar Borse, sir.
Good evening. I'm audible, madam?
Yes, sir, you are audible.
Yeah. Respected Dice, on behalf of Microbiological Society of India, Vidya Pratishthan Arts and Commerce College, it's my privilege to introduce one of my friend and academic colleague, Dr. Rebecca Thombre. Just to introduce her enriched biodata, Dr. Rebecca Thombre is a London-based senior scientist affiliated with Blue Marble Space Institute of Science, Washington, USA, and is a visiting scientist at the University of Kent, England. She is the recipient of the Europlanet European Union Award for Astrobiology Research in Spain and has traveled to the USA, UK, Europe, and Taiwan as a visiting scientist for research. She has completed her graduation and post-graduation in microbiology from University of Mumbai, India. She was the recipient of Eureka Forbes Award for the first rank in the Mumbai University. She has pursued her PhD in microbiology from Agarkar Research Institute, Pune. After her PhD, she pursued independent research in microbiology with projects funded by Europlanet, European Union, Indian Space Research Organization, that is ISRO, STC, SPPU, that is Savitribai Phule Pune University, UGC, National Synchrotron Radiation Research Center, Taiwan, and DCOD, Savitribai Phule Pune University. Over the period of 10 years, her research received funding of around 4.5 million Indian rupees, and she has filed patents and published more than 70 articles, 12 book chapters, and 2 books. Her research includes extremophiles in astrobiology and the formation of prebiotic molecules in impact shock. So this was an enriched work what Dr. Rebecca Thombre has been doing. During the academic interactions, I found her thinking out of the box, out of the world. And I think so, the research what she is pursuing, that is also out of the world. That is the space research what she is working upon. So with this brief introduction, it's our privilege and honor to have Dr. Rebecca Thombre for today's guest lecture. I now request Dr. Rebecca madam to deliver her speech. Yes, Devi madam. Yeah.
Yes, sir. Yes. Yes, it is indeed said by Albert Einstein, we owe a lot to Indians who taught us how to count, without which no worthwhile scientific discovery could have been made. So it's aptly said, ma'am, Indians are ruling the world across the globe. So we are proud to have you. Over to you, ma'am.
Thank you, Nilima ma'am and Borse sir, for the kind words. Yeah, can you see the slide? Is the slide visible?
Yes, it's just getting shared, ma'am. Yeah, it's visible.
Yeah. At the outset, I want to thank Dr. Arvind Deshmukh sir, Principal BP Shinde sir, Nilima Devi ma'am and Pendharkar ma'am for giving me this opportunity to share my passion for astrobiology and especially space microbiology with you all. So I have divided my talk into 3 sections. The first part, I'll give you a brief introduction about astrobiology and how space microbiology fits in the interdisciplinary field of astrobiology. I'll then be sharing a little bit about my research in extremophiles and origin of life, a part of research that I've done in India and a part of research that I'm doing over here. in the UK. And then I'll be sharing a few scholarship opportunities for students who wish to pursue their research or who wish to pursue their PhD or postdoc in the United Kingdom. So to start with, I think this month everybody saw the touchdown of the Perseverance rover, which was sent by NASA. So this is extremely interesting and exciting time for astrobiologists and space microbiologists in the entire world, because a lot of missions have been sent to Mars and a number of missions are going to be sent in the next 10 years. So astrobiology is an interdisciplinary field of science which explores the origin, the evolution, and the distribution of life in the universe. This is based on three very important tenets. The first is, where do we come from? That is, how exactly life began on the surface of Earth. Second, are we alone in the universe? So is there presence of life beyond Earth? Are there habitable zones like Mars or Europa beyond Earth? And the third one is, where are we going? What is the future of life beyond Earth? Now, why study astrobiology? What is the significance of this field? Now, the simple question is, even today, despite all the advances we have in science, in medical science, and in spite of the research that we've done, till now, we have not been able to answer a basic question. That is, how did life exactly originate on the surface of Earth? Today, we have the COVID pandemic, the entire world is in lockdown. Even today, we are not able to answer simple questions related to evolution of viruses. Did bacteria come first? Did viruses come first? So what we know from fossil records is that the Earth was formed somewhere around 4.5 billion years ago. And we had a stable hydrosphere, that is formation of water on the surface of Earth, 4.2 billion years ago. But the first cells of life, that is the first responses of living beings on the surface of Earth, was somewhere around 3.5 billion years ago. So between 4.2 to 3.5 BYA, there were certain events which took place. There were right ingredients of life, which is, of course, the first is water. And then you have organic compounds that would be amino acids, or you would have other bases. And then you had the right conditions, which led to the formation of the first cells. Now, there are a number of theories which try to explain how exactly the first cells are formed. You have the lithopanspermia theory, which is also called as the Fred Hoyle's theory. You might also have heard about the Hoyle-Narlikar theory, which says that the ingredients of life were distributed in space, and probably meteorites or asteroids carried these ingredients of life from outer space, extraterrestrial involvement of formation of life on Earth. You have the cosmozoic theory. I think all microbiologists might have studied theory of spontaneous generation and abiogenesis. So there are a lot of theories which are trying to explain whether RNA came first, DNA came first, how the cell macromolecules were formed. But even today, unfortunately, we have not been able to answer simple questions related to evolution of our first cells. All we have are theories. So how did we go from a planet which was completely inhabitable and inhospitable in 4.5 billion years ago to a planet right now which has more than 1 billion species? We still have not answered these questions. Now, why is microbiology important in astrobiology or in origin of life? What is the role that a microbiologist would play in this subject? Now, fossil records tell us that archaea are the most ancient organisms on Earth. Now, if you all have ever seen or if you all have ever visited Australia, you might have seen stromatolites at the Shark Bay. Now, stromatolites basically are microbial fossils, and the dating suggests that the first ancient organisms on the Earth were basically archaea or archaebacteria, which came around 3.8 million years ago. Unfortunately, archaea, who are the most ancient organisms or the ones who evolved the first on the surface of Earth, were not discovered until 1970s. Now, we all know that microorganisms were discovered far around in 16th century by Anton van Leeuwenhoek. However, it was only in 1970s that extremophilic archaea were discovered, And I highly recommend you all to read this groundbreaking publication of Carl Woos and George Fox, where he declared, or where he published about this third domain of life, which is archaea. Now, what is so interesting about archaea is that even today, a number of different archaea are still being discovered, and even today we have not completely understood the microbiology, the physiology, the biochemistry, the metabolism of this group of archaea. There are certain examples, or there are certain distinct organisms which have been studied, but we still do not know the entire metabolism or the methods of adaptations of these organisms, and how these organisms adapted themselves to primitive earth, and how these organisms might have evolved to the higher forms of organisms. Now, what we know about archaea is that they are polyextremophiles. They can survive high pH, high temperature, pressure, gravity, salt concentration, and they can probably survive in space-like conditions. One very interesting paper of Das Sarma et al. In 2006, which is published, says that halophilic archaea, or the archaea which survive in extremely high salt concentration, about 20% NaCl, are candidate exophiles. This was the paper which caught my attention around 10 years ago, and I started my research in this field in halophilic archaea and in studying about halophilic archaea, because this paper was one of the groundbreaking papers, especially for in my personal journey in research. So what is so unique about halophilic archaea? First of all, they require a minimum of 10% NaCl concentration for growth, and most of them grow, that is the extreme halophilic archaea, grow about 20 to 25% NaCl. Now, when they grow at such high salt concentration, they produce a pigment which is called as bacteriorubrin and a bacteriorhodopsin. Now, recently, you all must have read the research papers which have been published based on purple earth. So now these pigments are considered as life detection markers. So most of the sensors or systems which have been sent for detection of life in outer space, these two elements or these two pigments are going to be the key pigments which will be used for detection of life in outer space. So this is very important in terms of the pigment study of these organisms. Now, many of you all can ask this question: do extremophiles survive in space? Is it possible that extremophiles can travel in space, which is the lithopanspermia theory, or which is the panspermia theory? Now, exactly to test this, there was a biopan mission which was conducted by the European Space Agency, and they exposed this organism Halorubrum cavuta species for short term in space. And in the Expose-E mission, they exposed Halococcus dombrowski for 559 days. And it is extremely interesting that these halophilic archaea not only survive in outer space, but they can also thrive in outer space. Now, this is very important in terms of planetary protection. Does that mean that if we carry these organisms to outer space, does that mean that if we use these organisms, can we colonize Mars? So this is a very interesting new avenue of space microbiology research that has been going on about terraformation of Mars or colonization of Mars using organisms from Earth. Now, how are extremophiles important in Martian astrobiology? Well, 2 years ago, there was this paper which was published in the journal Science by Orosi et al. Now, MARSIS, which is a radar instrument on board the European Space Agency's Mars Express orbiter, they detected the traces of water on Mars. So it was confirmed that Mars now has water. Mars has atmosphere. Mars also has salt deposits, which are chloride deposits, which was detected by NASA's Mars Odyssey orbiter. So now Mars already has the right ingredients for evolution of life. Mars has water. Mars also has organics. Mars has an atmosphere which obviously has more of carbon dioxide and very less of oxygen or hardly any oxygen. But does that mean that if right conditions could be provided, life can evolve on Mars? Now, these are the questions which a number of missions have been trying to answer. Did you ever imagine, despite the COVID pandemic, despite the lockdown, NASA still sent the Perseverance rover to Mars, and a lot of research is currently underway in UK and European Space Agency for sending the Rosalind Franklin rover, which would probably go in 2023. So a number of missions, astrobiology and space microbiology missions, have been sent in Mars since the Vikings mission. That was the first mission in the 1970s and till now. And in the next years, again, a number of missions. We know that UAE has sent a mission. India, of course, sent a mission 2 years ago, and it is again preparing for the Mangalyaan 2 mission. China has sent missions. So a number of missions have been sent to Mars for checking or assessing the presence of life, either past or present. Now, it is not possible for us researchers to get a hand on samples or Martian regoliths or Martian rocks. So what we do is, in case we have to study research related to Mars, or if we have to study research related to Martian environment, we turn back to Earth and we try to find habitats or sites which are similar to sites present on Mars. So these are called as planetary field analog sites, or these are called as terrestrial analogs. Now, all over the world, there are a number of terrestrial analog sites. I was very fortunate that I received a travel grant and an award to go and study one of the sites. But if you see in India itself, we have 4 very important planetary field analog sites, which in some way or in other, that is in geochemistry, in the salt composition, probably in the entire makeup, in the paleosciences, bear some kind of resemblance to the sites which are present on Mars. So the first site over here which I've shown is the Lonar Lake, which is a meteorite impact crater. This again is a very common planetary field analog site for the craters which are present in Mars. Then you have salt deposits or salt mines, or these crystallization ponds, which has extremely high salt concentration. I just told you that Mars has got an extremely high salt concentration, around 20 to 25% NaCl, and very high perchlorate composition. So these sites can be used to study simulated Martian conditions. Then you have mud volcanoes in Andaman. Again, volcanism is a very common phenomenon on Mars. So mud volcanoes in Andamans can be used as planetary field analog sites of Mars. And then you have the salt lakes or the rock samples in the Himalaya, in Ladakh, in Leh. Again, these are very common planetary field analog sites. And in fact, we have written an entire chapter related to these planetary field analog sites. Recently, we published this in a book which was published by Elsevier. So what kind of studies can you do related to these planetary field analog sites? So I've just given you a brief overview of these are extremely simple physiology experiments, microbial biochemistry experiments that we have done in our laboratory in India. And all over the world, in fact, they do these experiments. So you basically identify a planetary field analog site, you isolate organisms from it, you identify it using our normal biochemical morphological identification and sequencing. And then what we do is we expose it to Martian stress or space-related stress. So Martian stress would be the salt concentration or the perchlorate concentration which would be present on Mars. So we mix or we have salt and perchlorate in Martian regolith. Martian regolith basically is the soil or sample which has similarity or which is a simulated Mars soil. Or you expose it to space-related stress. So space-related stress would be gravity, that is low gravity, which is called as microgravity. And after you expose your extremophiles to these conditions, what we do is we study the physiology, we study the antimicrobial resistance pattern, we study the proteome, we study the pigment production, we study the morphology, we study the intracellular ion accumulation. We basically study if the organism survives in these Martian conditions or space-related stress. And if it survives, what would be the mechanism of survival? Is there any change in the antimicrobial resistance pattern? So these are the basic physiology experiments that we perform when we do space microbiology studies. Now, this is just an example of the studies that we had done. We had collected samples from certain salt pan beds, isolated the organisms. We isolated around more than 100 organisms, deposited them in NCCS, deposited them in the Agarkar Research Institute's Culture Collection Center. And one very interesting research that I would like to share that we did is, in terms of microbial physiology, is when these organisms are exposed to high salt concentration, they tend to sequester sodium ions inside because you have sodium ions outside in excess. and less of sodium ions inside. But one of our research paper that we published, we proved that instead of sodium ions, these organisms sequester potassium ions, because potassium ions require less of energy, less of ATP, and it is thermodynamically more better. So this is one very small contribution that we have done in microbial physiology in terms of halophilic archaea. The second examples of studies that we have done are the microgravity studies, which we did in collaboration with the University of Pune's Biophysics Department with Vidyasagar Sir. And we expose these organisms to a variety of low gravity conditions, because on Earth, the gravity is 1g. And if you see on Mars, it is 1/3. In space, the gravity is extremely less. It is zero gravity. So we simulated similar conditions in the laboratory in University of Pune, and we exposed our organisms to low gravity, and we found that these organisms not only survive in the gravity, it also affects the physiology, and there is also change in the antimicrobial resistance pattern of these organisms. Now you might say, what is so important in changing of the antimicrobial resistance pattern? Now what happens is, when astronauts go in outer space, they are exposed to low gravity or zero gravity. And astronauts also have skin flora or microorganisms present on their skin, or you have the internal gut microbiome. When microorganisms are exposed to low gravity, they tend to become more resistant to antibiotics, and opportunistic pathogens basically increase their pathogenicity. And because of that, this can pose a very important health threat to the astronauts who are traveling for so many days in outer space. So this study, we published this research paper in one of the Springer journals. So this study was very important in my next proposal, wherein I submitted my proposal for studying astronaut health and microgravity. Now, we also studied what would be the chaperonin proteins which are produced in space-related stress. And this study we have done using MALDI-TOF MS. We've reported a number of proteins and again published in a number of papers related to the chaperonins which were produced in halophilic archaea and stress. We know the common chaperonins were HSP40, HSP60, HSP70, and small heat shock proteins. Now, when we talk about extremophiles, we tend to search, novel environments or novel niche habitats to isolate these microorganisms which are extreme. So we turned to rock microbiology, or we turned to rock samples that have been submerged in water, which have been underneath the sea, or which have been probably in Ladakh. And we tried to collect these samples and try to isolate organisms from the center of these rock samples. And since these rocks are porous, they have a very high concentration of salt, or they have a very high concentration of ions in the center. So it is most likely that we can isolate extreme organisms from such rock samples. So we have isolated a number of different organisms. Example, Bacillus thermocopri. This is from a meteorite impact crater from Rajasthan. Kosuria rosea is a psychrophile which is isolated from a Himalayan rock. So this rock was collected from Ladakh. Then we have Halomonas, which we've isolated from halite samples from Spain. And then this work was in collaboration with Sauche sir, where we've isolated a number of alkalophilic and halophilic organisms, Halovivax and Nitronococcus, again from these laterite rock samples. So shifting gears to isolation of extremophiles from rock samples to isolation of extremophiles from analog sites, I had an opportunity under the Europlanet program to visit similar planetary field analog sites which are present in Spain. So in Spain, you have a number of sites which have extremely high salt concentration. So these are basically salt lakes, they are lagoons, but they also have extremely high concentration of sulfate. They're highly sulfated lakes. So there are two such systems which are called as Terrace Lake system and the Pennaveca Lake system. So I'll be showing just about one of the lake system or one lagoon system that I visited. So if you can see over here, this is an entire pink color, reddish pink color lagoon, which is called as Laguna de Pennaveca. If you see beyond this lagoon, the entire water of that system is green in color, but it's only this lagoon which has turned bright pink, bright reddish pink in color. So I traveled with my colleague Felipe Gomez, who's from the Institute of Astrobiology at Spain from Madrid. And we traveled with a mobile laboratory over there to the site. We could collect the samples, we could process the samples. And we were really excited to study what would be the organisms which give this lake this bright pink color. We also use these organisms to do some astrobiology and space microbiology related experiments. So we published our research work at the European Planetary Science Congress in Berlin, and it was really received very well. We reported Danielella salina. This strain is a novel strain that we reported for the first time. We called it EP1, EP for Europlanet, which is a funding agency, which gave this lake its bright pink color, along with a number of halophilic archaea, which again we have isolated and we have studied, especially over here in Europe. There was a very big press conference after our presentation at Berlin, and our research was covered in more than 20 languages all over Europe, even in China, in Russia, in different European countries, and even in India. Nature India wrote a special article related to our studies in Europe. So it was really very inspiring and motivating for us that our research was getting covered, and space microbiology was slowly making a niche habitat, especially in India as well. So there are similar such lakes in Australia. This is an example of the Lake Hillier, which is there in Australia. And I was very excited when last year we had the Lonar Lake turn pink in color. And I was very excited, as well as I was very upset because I was not in India at the moment. And I really wanted to know what would be the reason for this entire lake to turn pink. And I think later there was a research article, there was a news article which I read that it was these halophilic archaea because of which the entire Lonar Lake turned pink in color. Shifting gears to a little bit about meteorites, I have done a lot of research, especially in meteorites and prebiotic chemistry. But since today's lecture is on space microbiology, I won't spend a lot of time in prebiotic chemistry and meteorites. But I'd just like to share that in the beginning of my talk, I had shared that you need the right compounds, especially for formation of cells, or especially for origin of life. And there is one theory that probably meteorites could be the sources of building blocks of life. So there is a group of meteorite which is called as carbonaceous chondrites, which contain amino acids. And in fact, we have published a paper because when there was a meteorite fall in India, I had the chance to get the sample of the meteorite. I was a member of the team which visited the site, and we reported more than 13 amino acids from the meteorite that we collected. So meteorites are very important in astrobiology and space microbiology for two reasons. The first is it is said that they are precursors, or they carried packets of amino acids when in prebiotic earth, when the cells were forming. So probably the source of amino acids which formed the first cells when our cell was evolving were extraterrestrial. And second, there is this very interesting paper which was published in Science years ago by David Mackay et al. Again, I would suggest students to have a look and read this paper. So Allen Hills is a Martian meteorite. So it's a Martian rock. This rock came from Mars, and it was found in Allen Hills in Antarctica. So there are different types of Martian meteorites. You have Shergottites, you have Nachlites, and I was very lucky that I could see these meteorites in person at the National History Museum in London. So this is an image which was published in the paper then. So this group studied this Allen Hills meteorite, and they found fossil remains of past Martian biota. So I would highly suggest you all to have a look at this paper. And after this paper was published, NASA and a number of space agencies all over the world started preparing for missions, because now they had got a meteorite which has some fossil remains of probable Martian life. So it is very important for us to collect samples on the surface of Mars and actually understand that was there a possibility of life on Mars previously when it had water and it had organics. If so, what happened now that the entire life has been obliterated? Is it possible that in future, if we do not take care of Earth properly, is it possible that Earth also can face a similar situation as the situation on Mars? So meteorites and studying these meteorites is very important, especially in terms of astrobiology. A small experiment that we performed when we were in India was there was a meteorite impact which happened in Rajasthan. It is called as Mukundpura meteorite impact crater, and I was very lucky to be a part of the team to go there to collect the samples over there, to collect the meteorite also. So from the meteorite impact crater, fresh meteorite impact crater, we collected certain rocks and samples, and we isolated an organism which is Bacillus thermocoprae, which survived impact. Now, whenever a meteorite falls on the surface of Earth, it falls with such high velocity, such high impact, that it produces shock waves, and it obliterates the entire microbial diversity of the region. It is not possible for microorganisms to survive impact conditions. However, the organism that we isolate survived this impact, and what we did is we simulated the similar impact conditions in an impact shock tube. So we worked with Physical Research Laboratory, which is in Ahmedabad, and we've also done impact research experiments with the Indian Institute of Science in the Hypervelocity Lab Center, where what we have done is we have exposed prebiotic molecules as well as these organisms to impact shock. So we simulated meteorite impact conditions and we tried to see if the organism survives. And to our surprise, the organism did survive. And in fact, we have published this research paper in the journal Astrobiology. Now, what is so interesting is that since microorganisms can survive in space, microorganisms can survive meteorite impact, is it possible that you can have transfer of microorganisms between planet, which is called as interplanetary transfer? Is it possible that via lithopanspermia or via panspermia, microorganisms or spores can travel from one planet to other? So these are the questions which we are trying to answer in space microbiology. Moving ahead, well, I get asked a lot of questions from students as to what subject should I take to pursue astrobiology? Should I take geology? Should I take engineering? Should I take astrophysics? So astrobiology basically is an umbrella of interdisciplinary science. I think as of now, it is one of the most interdisciplinary sciences that we know of. So you can take geology, geochemistry. So we have a group of scientists who are geologists, who work on fossils, who work on rock samples, and then they try to date these samples and try to find evidences of origin of life. Then you have an entire group which pursues astrophysics or astrochemistry, astronomy. So these would be graduates from physics, from chemistry, from mathematics who contribute in astrobiology. I think one of the major chunk of people who contribute in astrobiology are engineers, who again build spacecrafts, who build sensors, who build systems, who build payloads for these astrobiology experiments, especially experiments which are conducted in space. And then we also have a group of artists, painters, philosophers, policymakers. I won't call them as a non-scientific group. This is basically a group in arts and humanities who also play a very important role in astrobiology. And finally, in space biology, we have different subjects which form a part of the space biology specialization, which comes under astrobiology. So the first one over here is space microbiology. And I'm very happy to hear that I think in entire India, probably Pune University would be one of the first universities. I've heard that they are planning to introduce a module on space microbiology in their syllabi. So, which is very important because the next 10-20 years, this subject is really going to have a lot of demand in future. The second and the most important subject is space medicine or human physiology, where especially we have this center in King's College in London, where you have doctors who are doing MBBS, or you have scientists who are working as to how space can affect human physiology. and this is extremely important for research related to astronauts. Then you also have a number of botanists or people from plant sciences who are working in space farming, and they're trying to study tissue culture in space. Of course, you have stem cell and tissue regeneration in space, because a lot of research is being done to study how space can affect bone regeneration, or how space can affect the tissues of human beings. A very interesting, again, new frontier in this field is again insect astronauts. It is also called as astroentomology, wherein you have a group of scientists who study how fruit flies, that is Drosophila or silkworm, moths. You also have tardigrades. They try to study how these higher forms of life can survive in space. Does it have any effect on their DNA? Do they have any mutations? Does it affect the phenotype? And then one of the novel or one of the new field is synthetic biology in space, wherein again a lot of research has been done, especially in NASA Ames, related to synthetic biology in space. So overall, I just wanted to show how space microbiology is just one small subdivision or subsection in space biology, which comes under the entire big umbrella of astrobiology. Um, so moving again to space microbiology, I just wanted to share some of the topics or the frontiers in space microbiology, the kind of research which has been done, and the kind of research which is happening all over the world. So of course, the first and the most important thing is exploring and searching microbial life in space or in habitable zones. So we had the Mars Vikings mission, which was in the 1970s, and this year we had the Mars 2020 Perseverance rover. We have the Europa Clipper mission again, which is going in 2023. A number of missions are going to roll out in this next decade related to space microbiology. Another very interesting thing is to study microbial diversity in the International Space Station. You all must have heard about the isolation of the new organism Solomibacillus calami, which was named after our President Abdul Kalam sir, which was again a microorganism which was isolated from the International Space Station, and then studying microbial diversity in spacecrafts. This topic is very important, that we study microbial diversity in spacecrafts, because whenever we send spacecrafts or rovers or landers in outer space, what happens is when these systems land, since they're powered by nuclear energy, there's a lot of heat that is dissipated. Now, say for example, if they land in a spot in Mars and a lot of heat is generated, so you're going to have high temperature, and by mistake, if any microorganism from Earth hitchhikes, these are called as hitchhikers, hitchhikes via the spacecraft, this organism can be seeded on Mars, and then we do not know how this organism will evolve over there. And this is very important for something which is called as planetary protection. In fact, there is something which is in COSPAR, that is Committee of Space Research. They have an entire policy on planetary protection. So it is compulsory that for every space organization, you should have a division of planetary protection, where you should have microbiologists who are working to make sure that the spacecrafts which go in outer space, or the systems which you're sending in outer space, are completely free from microbial contamination, so that we inhibit, or we do not have forward contamination or backward contamination. Unfortunately, in India, in the Indian Space Research Organization, till today, we do not have a planetary protection officer, we do not have a planetary protection cell, and we do not know how they comply with the planetary protection policies, or with the COSPAR policies, which were formulated with United Nations and COSPAR. So this is again a very interesting field, where you have a lot of microbiologists all over the world working and contributing in planetary protection. The next is again studying the effect of space-related stress on microorganisms. Some simple experiments which you can do in your laboratory, or you can do laboratory simulations. So when I visited the Institute of Astrobiology in Spain, they had a Mars simulator. So you have certain instruments which are called as simulators, in which you can simulate the atmosphere of Mars, you can simulate the soil composition, that is a regolith, and then you can expose your samples to such conditions and then see the response of terrestrial or earth-based organisms to Mars. And then, of course, you can have planetary field analog sites or terrestrial analog sites. When I was trying to study astrobiology and lunar lake, I saw that a number of papers were being published on by scientists who are foreign scientists. So I highly urge and highly encourage all the Indian scientists to take control, or to go ahead and explore the planetary analog sites which are there in India. Because what happens is, most of the times you have foreign scientists who come to India, they collect the samples, take the samples away with them, and most of our planetary analog sites get contaminated. So I especially encourage all the Indian scientists to explore these sites, because not only they are biodiversity hotspots, they should also be protected. Like you have UNESCO heritage sites, they should also be protected as sites which are planetary analog sites, because once they're contaminated, you cannot have such sites again. Then of course you have a lot of extremophile research which happens in space microbiology. You have ground based studies which are earth based studies, and one very interesting field of study is antimicrobial resistance in space, because this has a direct effect on astronaut health. So I hope I have been able to share about how space microbiology, or how microbiologists play a very important role, especially in astrobiology, and especially in this new field of space microbiology. Now, since I was talking with the professors initially, I thought it is a very good idea to share with the students about the scholarships which are available for Indian students in the United Kingdom. And I've highlighted 4 fellowships over here, because I know 4 of my students, or 4 of the people whom I know personally, have already received this scholarship. So for sure, if you can prepare, if the lockdown has given you enough time, you can spend time in preparing your applications if you're interested to pursue your higher studies or your research in the United Kingdom. So you can either come for your MS or your PhD, or you can come for postdoc. And for professors, there is a visiting a professor award, or a visiting scholar award which is there, which they can write to. I think most of them would be available near the nearest British Council. But what I'll also do is I'll share these details with the professors, Pendharkar ma'am and Nilima ma'am, so that she can circulate with the students. The British Evening Scholarship and the Rhodes Scholarship is the most common scholarship which you all might have known earlier. But the Betty and Donald Keating Scholarship is a very good scholarship, especially for students who are extremely bright and come from disadvantaged or less privileged background. So you all can spend an entire semester in Oxford University in St. Catherine's College and study subjects of your choice, and your entire airfare, your visa, everything is covered, and you can spend from 1 to 3 months at Oxford University studying a course of your choice. So this is a very interesting and a very good scholarship which you can apply. Then again, there are a number of scholarships. You have the Gates Scholarship, the Edinburgh Scholarship, you have the Marie Curie Scholarship, and for women scientists, there is a scholarship which is called as the Dorothy Hodgkin Scholarship, which is the Royal Society Scholarship. So these are the scholarships which Indian students can use for pursuing the MS, PhD, or postdoc in the United Kingdom. So to summarize, I try to share a little bit about astrobiology, and I hope I have been able to share about how space microbiology fits into astrobiology, which is an interdisciplinary subject. I shared a little bit about my research of extremophiles, and on purpose I shared about my research which I have done in India, so that you all also know that it is very easy to take up small projects, make small beginnings, tiny steps, and then you can proceed towards larger projects in space microbiology and astrobiology. And I've tried to share a few career opportunities for students, the scholarships which are there. In case if you have any further queries regarding career opportunities, I've just listed my email address, and I'll be more than happy to go ahead and share any information if needed. Before I end, I have to thank all my funding agencies, especially the Space Technology Cell of Pune University and ISRO, because they funded my research from 2013 to 2018 or 2018 India. So I got a funding of two large projects from ISRO, which was very helpful and useful for my research. Of course, BCOD, literally, I think 10 years ago, 10 years ago, they funded my research on bacterioruberin, Europlanet, European Union for funding my research in Spain, in Europe, UGC, and of course, all my collaborators who have helped me, who have collaborated with me for my research. So I hope I have been able to, ignite a little bit of curiosity in this field of space microbiology and astrobiology. And I would like to end by a quote by Arthur Clarke. Two possibilities exist. Either we are alone in the universe or we are not. And both are equally terrifying. So thank you for giving me this opportunity. And I will turn over to the organizers for more. Yeah.
Yes, ma'am, it was indeed a complete view from rock science to the space microbiology after those official proceedings, which might have little bored the students, but after that, it was such a refreshing talk of yours, which had literally taken the students to visit the entire space from Mars to all. Thank you, a big thank you to you, ma'am. Yes, it was indeed a great plethora of the entire space science before the students, which I think is one of the areas. It is called extraterrestrial, but it is also for the students a little beyond imagination as one of the area where they should look forward to. So it was indeed a great pleasure hearing to you, ma'am. Yes, you started your career in Pune, but you could move on to such a great extent of the space microbiology, which again we are so fortunate that through our international webinar we could bring it back in a very small tip of the iceberg, maybe I would like to say, in front of the students, making them at least to make a very small beginning. As you rightly said, the tiny steps lead to a great achieving moment for the life of the students.